Hello students, welcome to this session. This is the second session of chapter 13. In this chapter we are studying regarding photosynthesis in higher plants. During last session we have started the history of study of mechanism of photosynthesis. How did the world come to know regarding the complete mechanism of the photosynthesis we are discussing? To know the mechanism of photosynthesis is not a single day story. It has taken 200 years time to know the complete mechanism of photosynthesis. In last session we have seen how the Joseph Priestley discovered the oxygen gas in 1774. He propounded that green plant release oxygen and that oxygen compensate or restore the damaged air which was damaged by the breathing of living organisms and burning candle. Actually, the name of oxygen gas was coined by another scientist, Lavoisier. And when the oxygen gas or this fact that green plant release the oxygen, the world came to know this fact. Contemporary scientist of Joseph Priestley started thinking at a point that during combustion reaction, carbon dioxide and water molecules are released and a certain amount of energy is also released during combustion. They thought that during combustion, oxygen combines with carbon and hydrogen atoms respectively, and during this combination energy is released. Then what about the reverse reaction? There was confusion among those scientists. Reverse means if separation of the oxygen molecules is taking place, separation from hydrogen and carbon is taking place, then certainly that reaction absorb energy or that reaction require energy to separate the oxygen atom respectively from hydrogen and carbon. This doubt was cleared by a scientist, namely Jean Ingenhousz in 1779. By his experiment, he propounded that evolution of oxygen gas by the green plant is light dependent. Only in presence of sunlight, the green plant release oxygen gas. He arranged an apparatus like this figure. He took a beaker, a funnel, and a test tube and put water in beaker and put some hydrilla plant. Hydrilla plants are aquatic plant. Hydrilla plants. And covered those hydrilla plant with funnel. And on the narrow end of the funnel, he put inverted water-filled test tube. And firstly, he put this apparatus in dark. He found no change there. When he put the same apparatus, this arrangement, in sunlight, he observed number of bubbles are produced from the hydrilla plant. And those bubbles were releasing or containing a gas. Later on he tested that gas was nothing but oxygen gas. Oxygen gas. And by this way, he propounded that the evolution of oxygen gas by the green plants is sunlight dependent, light dependent. Only in presence of sunlight, the green plant may release oxygen gas. Later on, this hypothesis, this information of Jan Ingelhouse established a landmark in the field of the study of photosynthesis. The scientist came to know that the photosynthesis is light dependent, and during photosynthesis oxygen gas is released, and by this way they found the information of the Jan Ingelhouse true. And similarly, in the early 20th century, there was a scientist, namely T.W. Engelmann. T.W. Engelmann, in early period of the 20th century. He also established the relationship between light and photosynthesis. By his own experiment, he detected the points on the green plant, maximum rate of photosynthesis. For his experiment, he took a prism and a jar. In glass jar, he put certain lower plant, a type of alga, that is cladophora. He put cladophora in suspensions of aerobic bacteria. And thereafter, he passed the sun rays through prism. After dispersion of light, you know the dispersion of light after passing through a prism, dispersion takes place and the sun rays divides into 7 color. Vibgyor. 7 color. Violet, indigo, blue, green, yellow, orange and red. After passing the 7 colors of the light on the cladophora, he observed that the aerobic bacteria which were around the cladophora started accumulating on the points or region where the red and blue color of the light was falling. Blue and red color. These color of the light are more important, more effective for the mechanism of photosynthesis. He observed because there was excess evolution of oxygen, maximum evolution of the oxygens was those regions, those points where the red and blue color of the lights was falling. That was the reason why the aerobic bacteria started accumulating on the regions of the points where red and blue color of the light were falling. And this information of T.W. Engelmann was very important. And this spectrum test to know the effectiveness of the color of the light is called as axiom spectrum. This experiment is popularly known as axiom spectrum test or experiment of the Engelmann. And that was the first axiom spectrum described by the Engelmann. And later on, the absorption spectrums according to the type of chlorophylls was discovered. More or less, the action spectrums and absorption spectrums resemble each other. Now we will discuss the certain other experiment done by other scientists in the field of the study of photosynthesis. In 1885, there was a scientist, namely Julius von Sachs. With his experiment, he propounded, he informed the world that in presence of sunlight, green component of the plant cell takes part in photosynthesis. Green part or green component, later on that component is called as chloroplast. That was very important information given by Julius von Sachs. By his experiment, he informed the world that photosynthesis only takes part in green component, green cell organelles found in cells of plants. Later on, after him, that cell organelle or that component of the cell of the plants is called as chloroplast. He also informed that after photosynthesis, the food produced is nothing but carbohydrate, and that carbohydrate is stored in the chloroplast, firstly in the form of starch. That was very important information, because after getting this information of the Julius von Sachs, the scientists started testing the leaves and other parts of the plants to test the formation of the starch, to know the potency, to know the mechanism of photosynthesis. And that test is called as starch test. Starch test. Certainly in previous classes you have studied regarding starch test. The starch test is based on the information of the Julius von Sachs. For starch test, firstly we do the plants de-starched, keeping the plants, green plants, in the dark for some days. You may also test to know the photosynthesis is light dependent. You take a plant and firstly you discharge the plant, and thereafter you cover the certain part of the leaf by a black paper. After that you put the plant in sunlight for some hour. When you will test the leaf for starch formation, you will find that the part where the leaf is covered by black paper, the starch is not formed. The starch is not produced. Why? Because the part which was covered by the black paper did not get the sun rays. So, in that portion of the leaf, the starch is not produced. But in remaining part, you will get the production of starch. This experiment proves that photosynthesis is sunlight dependent, and this experiment is based on the information of the Julius von Sachs. Now, by this way, in the course of his study of photosynthesis, in middle of the 19th century, more or less scientists came to know the actual mechanism, complete mechanism of the photosynthesis. And on the basis of gathered information of the different scientists. In middle of the 19th century, the scientist gave the empirical formula of the photosynthesis. CO2 plus H2O, light, and C2O, CH2O. That is the empirical formula of carbohydrate, and oxygen is involved. This empirical formula was given in middle period of the 19th century. And after some time, in 1931, a landmark was established by a great scientist, a great microbiologist, namely C.B. von Niel. He did an experiment with purple and green bacteria. Green bacteria. C.B. von Niel, after doing the experiment with the purple and green bacteria, informed the world that when the carbon dioxide combines with certain suitable oxidative substance like hydrogen sulfide and water, in presence of sunlight in the green part of the plant, or in the case of purple and green bacteria, carbohydrates are formed. And accordingly, either the oxygen gas or certain other type of the gas is produced there. But after biochemical reaction, the hydrogen separated from those oxidative substances and which reduce the carbon dioxide into carbohydrate. In the case of purple and green sulfur bacteria, sulfur, the oxidative substance is hydrogen sulfide. Hydrogen sulfide combines with carbon dioxide, and here the CH2O carbohydrate is formed, and sulfur gas is released. Sulfur gas is released. Similarly, in the case of the higher plant, the water combines with carbon dioxide and CH2O and oxygen gas is produced. By this way, the C.B. von Niel cleared the confusion of the contemporary scientists that oxygen during the photosynthesis is not coming from the breakdown of the carbon dioxide, but due to the breakdown of the water. Later on, it was found that during Photolysis, the water splits at the site of photosynthesis and oxygen molecules, oxygen atoms are released and hydrogen atoms are also released side by side, which hydrogen atoms combines with the reducing agents and which are onward utilized to reduce the carbon dioxide into carbohydrate. So the information given by C.V. van Niel was very important and proved a landmark in the field of the study of photosynthesis. And after him, there was another scientist, Robert Hill, Robert Hill, who by radio isotopic technique proved this information given by C.V. van Niel, that water splits and is responsible for the production of oxygen during photosynthesis, and the hydrogen atoms are on the other hand responsible for reducing the carbon dioxide into carbohydrate. After giving this information, the Robert Hill gave the complete formula, simple complete formula of the photosynthesis. That formula is well known and well accepted today. That is CO2 plus H2O, sunlight, chlorophyll, C6H12O6 plus H2O, H2O plus O2. This is the complete simple formula which was given by Robert Hill by this experiment, radioisotopic technique, 6 year also, which are well accepted still now today. And by this way, the world came to know the complete mechanism of the photosynthesis, that during the photosynthesis, CO2 combines with water molecules, and here the carbohydrate is produced in the green part of the plant in presence of sunlight, and the photosynthesis is sunlight dependent, and water and oxygens are the byproduct. Here the main product is the carbohydrate. This is the chemical energy. Here the solar energy in the form of photon, is converted into chemical energy by combining these raw materials. And these are the overall history of the photosynthesis. Now in next session, we will discuss the site of photosynthesis, that is the chloroplast. We will firstly discuss the structure of the chloroplast in concise way, because in previous chapter number 8, we have already discussed regarding the chloroplast in detail. In next session, we will discuss the structure of the chloroplast and absorption spectrum in context of the different type of the chlorophylls which take part in the photosynthesis. Thereafter, we will discuss the different type of photosystem. You students continue with us. You watch the videos available on our institute, subscribe our channel, get benefited as well as help the institute in promotion of the YouTube channel. Thank you very much.